Thank you. So welcome everybody. It's a really great pleasure to welcome Dr. Gary Bader today. He is at the University of Toronto. He's a professor in the Donnelly Center and an international leader in computational biology. He has an undergraduate degree from McGill in biochemistry and a PhD at the University of Toronto with Chris Hogue at the Samuel Lunenfeld Research Institute and in the Department of Biochemistry, and did postdoctoral work at Memorial Sloan Kettering with a group of Chris Sander. And he's been doing so many interesting things. I hear about his work from time to time in all kinds of different areas in computational biology. And today, he's talking about cell diversity in development. And so please go ahead, Gary.
Great. Thank you very much for the introduction and for inviting me here. As Celia mentioned, I went to McGill, so unfortunately I'm not visiting in Montreal in person because I really like Montreal, but another time. So I'm going to talk about this question that we have about how cell diversity is generated during development, and I'll just acknowledge Maria Abuchakra. First off, this is her work that I'll be presenting, and I'm happy to see that she's in the audience, and so I'll hopefully be able to also answer questions as they come up. So I just want to as a general research background, our lab focuses on systems biology and mostly computational systems biology. Traditionally, we've been focused on understanding the genotype to phenotype relationship via understanding systems, that the genotype encodes systems, and systems explain phenotype. So by understanding systems, we are better able to understand the genotype to phenotype relationship. We've also developed a lot of community resources like Cytoscape and focused on network visualization and analysis and genomic data interpretation. But today I'm going to talk about stem cells. So a little while ago, over the past number of years, we've had increased interactions with colleagues in Toronto around stem cells in 3 different areas. So sort of the traditional way people think about stem cells is in development. That's, the original idea, I guess, comes from development. But also people like John Dick and Peter Dirks have figured out that there are stem-like cells in cancer, and they seem to be the cells that are both, on the one hand, untreatable because they act differently than the rest of the cells, and also responsible for growing the tumor. So a single cancer stem cell can reconstitute an entire tumor in another, in a mouse, for instance. And then also in the context of regeneration, there's a large project in Toronto, Medicine by Design, also collaborators like Peter Zanstra and Penny Gilbert. For instance, we've talked a lot about the idea of using stem cells for therapeutic applications. So not just stem cells, but just the idea of generating new tissue to repair damaged tissue. And so, all these 3 areas kind of converged on trying to figure out better understand the biology of stem cells and how they work in different environments. And so one of the first things that we wanted to try and do was try and develop a simulation model of a cell that's growing. And Maria developed this, originally this 3D bubble-based simulation that can grow cells. And our original idea was that we would start with a single cell and have the cells grow until they reached kind of like a ball state, which is kind of analogous to very early development. And then we sort of see if we could use this to better understand these systems. And so, Maria implemented this system, and we had a number of parameters that we defined. But we soon realized that we didn't really have enough information to define the system as detailed as we would have liked. In particular, some of the parameters that we were looking at were missing in the literature when we searched. So I'll talk more about one of those parameters and how we've been exploring it, but just to introduce the topic more generally, we know that every cell in our body maintains the same genome, but it has very different phenotypes depending on how what state or cell type it's in. So we know a lot about these cell types, and we're learning more about them every day, especially with in the era of single-cell genomics. But we still don't understand how these cell types really arise from early stages. So we know that there are lots of possible mechanisms that can influence cell diversification, intrinsic and extrinsic factors, environmental signals, and intrinsic factors like the epigenome, all sorts of regulatory systems, cell communication. Cells can communicate through mechano-signal transduction, for instance. Chromatin state we know is important. Gene transcription is definitely important. So we have lots of ideas about different mechanisms, but how do they fundamentally help cells make decisions about what they should do next? So one of the parameters that we sort of didn't have enough information about was the cell cycle. We know that it's in our mathematical model. We know that the cell cycle is fundamental to development, and canonically, what you learn when you look at the textbook is that the cell cycle has 4 stages, and it proceeds in a clockwork-like fashion. But as soon as we started looking at trying to get information about the cell cycle, like the duration of the cell cycle, we realized quickly that the cell cycle duration is variable and very context-specific. And this was, the earliest paper I think that we were able to find, and Maria was able to find to show this was one from 1960, where this person had figured out that the cell cycle duration was different in different tissues in a plant or an animal, and so that kind of makes the situation a lot more complicated. We also know that the cell cycle lengthens over development. So this is a plot of different measurements of cell cycle duration at different embryonic stages or days in mouse brain development. And over time, that's basically a general trend that the cell cycle starts off very fast presumably to generate a lot of different cells, and then slows down over time as the tissue reaches its mature size and volume and shape. And we also know that in fruit fly and C. elegans, the cell cycle duration can be very short. It can be as short as 8 to 10 minutes in early development. So that raises the interesting question. That raised the interesting question for us: is how do the genes get expressed? Because we know that the transcription rate is about 1.4, let's say, 1.5 kilobases per minute, up to maybe 3 kilobases per minute. So that's roughly, say, 100 kilobases of genome that's transcribed per hour. So long genes, there's a lot of long genes in the genome that cannot be transcribed in these time scales based on this rate. So that means that there's some kind of potential filter that's occurring based on the cell cycle duration, where if the cell cycle duration is too short, then longer genes can't be expressed. There's just not enough time for them to be expressed. And a gene, in this case, is the genomic gene. It's not the protein coding region. It's including introns and exons. And introns are almost, especially in multicellular animals, are really long. They're much longer than the exons, and so the vast majority of the genes are introns. So we definitely have to consider this whole thing, that they all get transcribed, and it takes a long time sometimes to transcribe these genes. So people have studied this over time, and for instance, we know that if a gene is, if a transcript is not finished by the time the cell needs to divide, then those transcripts are repressed. They might be degraded. And so the genes have to be expressed within a G1 phase, for instance, probably. So we also just did a sort of quick thought experiment. What would happen? What amount of which genes wouldn't be able to be transcribed if the cell cycle was short? So let's say we had the cell cycle defined as 1 hour, the duration of the cell cycle, and let's look at the distribution of gene lengths for all these different species. So this plot over here tells you the percent of the genome that would be restricted would not be able to be transcribed if the cell cycle was 1 hour. And you can see it's almost 20% for Homo sapiens genes. So there are some genes in the human genome that are like 2 million bases long. So that would mean that they need many, many hours to be transcribed. So we created a simple model of cellular development to explore how this relationship between cell cycle duration and gene transcription might have effects or might work. So the model is a simple mathematical model, it's agent-based model or rule-based model that defines a cell, and the cell has a genome. The genome has genes, which are introns and exons, so it's just the genome, the genomic positions of these genes, and those genes can be transcribed. And then cells can also divide according to a rule that just divides them after a certain amount of time, which is the cell cycle duration parameter. So each cell is assigned a cell cycle duration. So all of these are basically all the parameters of the model initially, the simple model. Then there's a rule that allows cells to divide, which basically takes the transcripts that are generated during the cell growth phase and randomly distributes them to the transcripts in the parent cell to the children cell. So this is sort of the one area that's random in the model. So we first asked how a transcriptional filter might affect transcript count. So just theoretically, again, sort of as a thought experiment, if we have a gene that's short, and so in this particular model, we usually numbered the genes 1, 2, and 3, etc., and the number corresponds to the length. So you could imagine that the gene 1 is 1 kilobase long, and gene 2 is 2 kilobases long, etc. But the length doesn't matter for this particular explanation. It's just that we have relatively short genes and relatively long genes. So if a gene is short, it can be transcribed if we have a cell cycle duration that's equal to 1, which is a cell cycle duration that allows gene 1 to be transcribed. And so we can see that we get expression of gene 1 and no expression of gene 2 and 3. If we set the cell cycle duration to 2 time periods, excuse me, we can now get 2 copies of gene 1 and 1 copy of gene 2. And similarly, if we make the cell cycle duration longer, we start allowing longer genes to be transcribed, but we get more of the short genes because they have more opportunity to be transcribed multiple times. So, so we can do this, we can run this like a bigger simulation of this using our model. So if we do thousands of runs of the model and have a lot more genes, we can look at how the cell cycle duration parameter affects the expression of genes based on their length. And our simulations show that short genes are expressed, as we expect, more highly than longer genes. Okay, so how does this compare to real data? There's a lot of single-cell RNA-seq data these days that we can make use of, and we made heavy use of trying to compare our model to what we observe in single-cell RNA-seq data. So, for instance, Briggs et al. Studied frog development and published their gene expression data, single-cell gene expression data, and if we look at the gene expression values based on the length of the gene, we can see that the pattern is the same as what we see from our simulations. So this doesn't prove that our simulations are correct. In fact, nothing that I'm going to show you today proves that our simulations are correct, but it doesn't disagree with the simulations. And so you know, we think that it's the simulations that this idea that we're proposing about a transcriptional filter based on cell cycle duration is a viable mechanism that we should study, because the data that we see doesn't disagree with it. Okay, so we can also look at this over many species. So there's a paper published in 2019 where they looked at, they measured single-cell transcriptomics for microglia across a number of different organisms. And in general, we see the same trend, which is that shorter genes are more expressed. Interestingly, the short genes are, you know, higher expression of short genes. It's not really being well described in the transcriptomics literature, although there was a paper about a year ago that said, hey, we found an important confounding factor in transcriptomics, that short genes are more expressed. And so, you know, we need to control for that if you're doing normalization. So anyway, just an interesting aside, but it's a simple idea, but it hasn't really been explored very much as far as we can tell. This top part of the plot just shows that the bin sizes for these, the thresholds that we selected to define short, medium, and long genes are not very different from each other. So there's no bias from that. So our model is, our mathematical model is single cell resolution, which means that every cell that we generate is tracked. And we can think about defining a transcriptome vector for each cell. So we can say that gene 1 has 2 copies, gene 2 has 1 copy, gene 3 has 0 copies, or, you know, we can define another transcriptome vector. And so we can think about our mathematical model as generating single cell transcriptomes. We can measure, and so we can use different measurements for how to study these transcriptome vectors. And there's a lot of analyses methods from single cell genomics or single cell transcriptomics that we can use to analyze the results of our simulations, as I'll tell you about in a second. But the simplest way we were thinking about this is that if we, for instance, have a genome in our model that contains 10 genes, and the shortest gene is size 1 and the longest gene is size 10, and we have this transcriptional filter mechanism working, which means that we've set a cell cycle duration parameter so that it's shorter than the longest gene. So some of the genes will not be able to be transcribed in some cell cycles. So if we set that cell cycle duration to a very low number, like it's a fast cell cycle duration, then there's not that much transcriptome diversity because only one gene, the shortest gene, will be able to be transcribed. So all of the transcriptome vectors will look the same. All the cells will just have that one gene transcribed. On the other hand, if we set the cell cycle duration to the maximum amount, we can all the genes can be transcribed, and due to our random process of distributing parent transcriptomes to children cell transcriptomes, we generate diversity, and you can measure that diversity in sort of the number of different vectors that we create. And then through the, if we simulate different values of this parameter, we can see that there's a clear trend that as we increase the cell cycle duration, there's more transcriptional diversity as measured by these transcriptome vectors. And so interestingly, so that's what happens with the transcriptional filter. Interestingly, if we don't have a filter, like if we set the genes to be very short, so that the longest gene is size 1, then that means that all the genes will be able to be transcribed at all values of the cell cycle duration. And we just quickly see that we saturate the diversity of the system, and so we just immediately generate a lot of different types of transcriptomes. And Maria figured out an analytical solution to this, so we can compute these. We don't need to, these values, we don't need to simulate it. But what was interesting for us in looking at this plot is that when we have a transcriptional filter, we have a controllable transcriptome diversity. So we can set the transcriptome diversity to any point along this line just based on changing the cell cycle duration. But when we don't have a transcriptional filter, which means that all the genes can be expressed, then we can't control that transcriptome diversity. It just maximizes that. It just gets all, generates diversity maximally right away. So we thought maybe that's an interesting tuning knob that's developing systems can use to choose transcriptional diversity, which might be useful. So that was one measure of transcriptional diversity where, I said basically every one of these vectors is considered different if there's any differences. It's not totally realistic, but it sort of represents maybe one side of one extreme case of what you might think of how to measure diversity of transcriptomes. The sort of standard way of doing this in single-cell transcriptomics, I'm sure most people know about single-cell transcriptomics. This is just an example of a liver single-cell transcriptome where we worked on, where each circle here is a cell, and the space here represents the space, the dimensionality reduced space of all the gene transcriptomes, all the cellular transcriptomes for each of these cells. And the distance between these points, the distance between these points correlates with the distance of the transcript of their transcriptome vectors. And so typically this is how people visualize and analyze single-cell transcriptomics. They cluster the cells based on their similarities, so they identify groups that represent, people have figured out that they represent different cell types, each group, and so we can use this to try and identify or measure diversity as well in our system. So in this particular case, we can see that the diversity of the cells in this tissue, there's about 20 different cell types based on the clustering that we've defined. We've defined 20 clusters. So maybe we can just use all of the standard technology and analysis for our data. And so when we do the same experiment that I mentioned earlier, we can see that if we change the cell cycle duration parameter when we have a transcriptional or a transcriptional filter active or inactive, this again is the same parameters that we used before on the gene length distribution and the number of genes in the genome. And the only difference here, by setting these, again, by setting the gene length distribution, we kind of turn the transcriptional filter on or off. And so when we have an inactive filter, we see that there's a certain number of clusters that gets generated, and it's not exactly the same each time, but roughly similar. But interestingly, when we have a transcriptional filter that's on, there's sort of, again, at the beginning of, if there's only one gene expressed, we don't get a lot of clusters. If all the genes expressed, they're very similar, the cells are very similar to each other, but in the middle, there's actually a lot of opportunity for clusters to develop. And again, even though it's a different pattern, it's a controllable, we see the same pattern that the transcriptional filter allows you to control the number of clusters that are getting generated based on the cell cycle duration parameter. So that was, the first experiment that sort of suggests that maybe the cell cycle duration can help control not just the number of transcript vectors that are created, but also might be able to create different clusters in the data, which in real single-cell RNA-seq data represents cells. So maybe the cell cycle duration parameter can also help you control the number of cells that are generated. And so that was that kind of idea. So we can also change things a little bit. So apologies, Maria, this is an old plot. We have an updated plot of this in our paper, which was published 6 months ago or so in eLife. But the idea here is that you can have in the lineage of cell divisions, you can you don't have to have the same cell cycle duration parameter everywhere. So for instance, you could have one branch that has the transcriptional filter on, and another branch that has the transcriptional filter off, and we see that, you know, we basically, if you do that, then you get a mix of outputs. So you can generate diversity if you want with one of the branches, and the other branch you can, for instance, have a very fast cell cycle duration, or like a short cell cycle duration, which basically means very fast cell cycle. So in this case, you would generate a lot of cells. So fast cell cycle duration generates a lot of cells. Slower cell cycle duration doesn't generate as many cells, but potentially could give you diversity. And so there's a little bit of a trade-off between the speed of the cell cycle generating lots of cells versus, you know, a faster cell cycle generating lots of cells, and a slower cell cycle generating fewer cells but more different types of cells. That's kind of the idea. And, you know, this is sort of the, like a single cell RNA-seq style plot of this, where we see, depending on the cell cycle duration, whether it's short here, you know, both branches basically have the same short cell cycle duration. But if we lengthen and so we get two clusters that are 50-50, sort of represented in terms of the gray and the blue lineage. If we increase the duration of one of these lineages, the cell cycle duration of the cells in one of these lineages, then we get a lot more diversity here, but we get fewer cells, and that trend continues if we make that continue to make that one longer. So that was what I was explaining before, just visually depicted here using the sort of standard way that single cell RNA-seq is analyzed. Okay, so I mentioned earlier that we know that cell cycle lengthens over development, this slide that I showed you earlier. So we can simulate that same case where the cell cycle duration is changing across cell divisions. And we had sort of a noisy way of doing this. It wasn't always exactly the same, but basically the average cell cycle duration increased over time. And so the pie charts here represent the same type of pie charts that you see here. So we have, in this case, a gray and a blue lineage, and they can each have their own cell cycle duration parameters. And you can see the pie chart measures the proportion of cells that's generated by the model once we set these parameters. So when we basically visualize those pie charts on this plot. So the parameters here are, you know, average cell cycle duration, and it's changing over time. And the pie charts show the proportion of cells that are in different, you know, basically lineages that have different cell cycle duration times. And you can see that we generate different patterns here, which is interesting. When we look at real data, we were involved in a study that's was published with Frida Miller in David Kaplan's lab. Scott Yezwa was the lead author. This paper was basically used single-cell transcriptomics to measure, to study mouse brain development, and we had 4 different time points of mouse brain development at different embryonic day stages of mouse brain development. And we also know from the literature what the average cell cycle duration is at these stages. It's been measured, and so we could kind of make the same plot with real data, and we can see that you know, as the average cell cycle duration increases over time, we get these pie charts that are different. And you know, the pie charts are not exactly the same, so we can't really say our simulation is recreating what we see in mouse brain development. But one of the things that we find is that that there are different proportions of cells that are generated, so the pie charts are different, and then there are you know, and it kind of works as you expect with the slower cells. The faster cells, yeah. So yeah, actually, now forget that point, but sorry. so the main take-home message is that the pie charts here, or the proportions, can be controlled. So we think that not just the cell diversity can be controlled, but we might also be able to control using cell division, using this type of system where you have an interaction between cell cycle duration and this transcriptional filter idea could potentially also be used to control the proportions of cells in the tissue as it grows. We tried to find as much data in the literature to kind of help us, I guess, check whether this model was looking like it matched real data. And we found, Maria found a couple of studies on Drosophila embryonic development. So this line here represents kind of Drosophila, larva, which is like a worm, and if you spread it out, you can think about it as just a cylinder. And one study had measured the expression of Hox genes. So you can see the different Hox genes are colored here based on where they're expressed. And these are the shapes here represent the genes, and their length is proportional to their actual genomic gene length. Another paper had basically measured cell cycle rates across these different parts of the embryo, Drosophila embryo, and we found that basically there is a correlation between whether there's a region here which is like a fast mitotic region versus a slow mitotic region that correlates to the length of the genes, the Hox gene family genes that are expressed. So in the fast mitotic region, we only see short Hox genes being expressed. In the slower mitotic region, we have longer Hox genes being expressed. And so again, this doesn't prove that our model is correct, but it doesn't disagree with it, and so by virtue somewhat supports it. And it's an interesting example. You might also think, okay, it's basically kind of interesting to sort of see how this development might work. If you have a tube that needs to grow, it looks like it's more efficient probably for the tube to grow at the ends, which is the fast mitotic regions here, than in the middle, which is the slower mitotic regions. And it probably makes sense. It's probably easier to push out from the ends than push the whole thing out from the middle. Okay, so we also found we kind of really struggled to kind of check whether this model is working. We tried to, one of the struggles we had with this project was trying to get some experimental data to support this, some new experimental data where we could actually change the cell cycle duration chemically, for instance, or change gene lengths genetically. And we just, even though we actually had some experiments running, they just didn't work out. And, so we were kind of trying to find all sorts of different ways to think about how this, we could kind of corroborate our findings. And one interesting thing that Maria found was, we thought maybe we can learn something about how long it takes different species to develop from a single cell stage to birth, basically, or some definition of birth. And Maria spent a lot of time collecting information about the developmental time for lots of different species, and then we can compare that to the median gene length of their genomes. And in general, there is a correlation, a pretty good correlation between how long it takes to develop, how long the organism takes to develop till quote-unquote birth, and how long their genes are in their genome. And the idea is that if it takes longer, if you really have to develop very quickly, like in a day or 2 days or 10 days, you can't really afford to have slow cell cycle duration, and so you probably won't be able to have a lot of long genes, and that definitely seems to be the case. We also looked at, if you just take all the genes in the genome, let's say the human genome, and you rank them by their length, their genomic length, and then you do a pathway analysis on those. Short genes and long genes participate in very different types of pathways. In fact, it's one of the strongest pathway enrichment signals I've ever seen. It's probably the strongest one I've ever seen. So long genes are very strongly enriched in mostly brain and muscle development. And short genes are enriched in core processes like environmental sensing, immune response, like defense, basically against core kind of transcriptional processes, and also things that are related to like the immune system and defending cells. So probably that's makes sense. We know that cell cycle duration can be quite long in tissues like the brain. It fits with our idea that longer cell cycle durations can basically allow you to create more cellular diversity, and the brain and the muscle are good examples of tissues in the body that have lots of cellular diversity. There's lots of different types of, especially the brain, but there's also lots of different types of muscle. On the other hand, for things that you just need to make sure you don't delay, like immune responses and core transcriptional machinery, a lot of those genes are very short. So this is, we also, just as an aside, we looked at other kingdoms of life. This is focused on multicellular animals, but we see in plants, for instance, they don't have long genes. So there's definitely there's a distribution of gene lengths in plants, but they're very, very short. And probably that relates to how plants might be more important for plants to respond to their environment because they don't really control their environment like animals can. You see the same thing in bacteria with very short genes, and single-cell organisms in general. And although this process could still play a role there, it's just the distributions are really shifted over to the short end of the gene length scale. Okay, so I have a couple of other things to mention, but the general conclusion is that we think that cell cycle, our model shows that cell cycle duration and gene length can be important factors in development. We can show by simulations, mathematical simulations, that they, or computational simulations, that they can be used to fine-tune the numbers of different types of cells, the proportion of those cells, and express gene expression programs, so it can act like as a gene expression switch in some sense. And so these are properties that are very valuable presumably in developing tissues, because developing tissues need ways of setting the number of cells that they need to generate and the number of different types of cells that they need to generate. And so, and there's a lot of cell cycle dynamics that changes over development, like I mentioned. So we think that this is an interesting area to further study to see how maybe that cell cycle duration is an important factor that controls tissue patterning and development and things like that. There's a couple of other things I want to mention. One is that sometimes people ask us, we kind of have an assumption about, this is just an extra point, I probably didn't mean to mention it, but we have an assumption in our model that basically the transcripts are generated, they're getting generated when as the cell grows. So we can choose to control whether the transcripts get inherited from the parent cell or not. And so we can also, sorry, I should have said, people know that in early development, there's this idea called the maternal zygotic transition. So the parent cell, the mother or the parent cell, in real animal development, provides a lot of the transcripts the zygote. And if the zygote's cycling quickly, the zygote can't transcribe all of its genes, but it's able to get some genes from its parent cell. And so this would change the distribution of the gene expression that we see. And in general, it would allow long genes to be occurring, even though they couldn't be generated from the beginning. And so this we can simulate this and show that our simulations can replicate that. And actually, I forgot to include here, there's some data that that people have published that shows that there's a strong bias in the types of transcripts that are inherited by the zygote are actually the long genes. And so this likely does occur in real single cell, really at the beginning of development. Okay, so that's the end of kind of the results that I wanted to discuss. One of the things that one of the general points I wanted to make is that my lab and in general our team is trying to think more about how tissues work, and tissues being systems of cells. So I mentioned at the beginning of the talk that my lab has traditionally been focused on systems biology, and in general, the past 20 years of systems biology have really focused on trying to understand cells. We have cell lines, cancer cell biology, cell atlas, everything's very cell oriented. And mostly it's about signal transduction pathways within cells, protein interactions inside cells, etc. On the other hand, physiology has spent 100 years at least thinking about how physiological systems work, but there's this kind of missing piece in the middle where we didn't have a lot of information traditionally, I think, about ways of measuring all the different cells at the tissue level. So now with single cell genomics, we can do that, and we're starting to get a ton of information about thousands or even millions of cells that are formed in tissues, and we can measure their transcriptomes and many other aspects about them. And this really provides the opportunity to apply all the systems biology concepts that we've developed over many years, thinking about how systems inside cell works to how systems inside tissues work at the scale of cellular systems. And so if we think about ecosystems themselves and how they work, and people have thought about ecosystems of cells before, mostly in bacterial, in the context of single cell organisms. It's a long history there, but in the context of multicellular animals that generate tissues, this hasn't been thought of as much because of the lack of data, I think. But we can think about how cells work together, what patterns are implemented, what mechanisms implement these patterns. So for instance, we find a lot of gradients in single cell transcriptomics data. How are those patterns implemented? They're probably implemented based on all the mechanisms I mentioned in the beginning of the talk, like cell-cell interactions and all the intrinsic and extrinsic factors that we know are potential part of the menu of controlling systems or regulatory systems in the biological systems that we're studying. But exactly how those work, we don't know a lot in terms of how they control tissue level patterns. So how are these patterns implemented and how do they work? How do cells change and evolve in response to the environment and internal programs? So there's a lot of interesting open questions that we can start addressing if we can figure out how to make use of all of these measurements that we have. And I think that what we tried to do here, as also a general point, was try to combine mathematical modeling or computational simulations with genomics. We know that genomics can make huge amounts of measurements, but it's hard to interpret. Mathematical models typically require very detailed measurements, which we don't have. Like we don't have a lot of measurements of cell cycle duration, although maybe we would get that with eventual single cell genomic technologies that are image-based, for instance. But if we can somehow try to think about how to use all the measurements that we're making in genomics to help parameterize or reason about models that we're developing on the computational side, maybe that can help us move forward and both generate questions for thinking about the genomics data and trying to validate models that we generate on the computational and mathematical side. Okay, so with that, I'd like to thank, I thanked Maria at the beginning, Ruth Isserlin, Finn, and Brendan also helped with this work. And we had a number of collaborators who focus on the developmental biology, stem cell biology in the context of development, cancer, and regeneration that we discussed this project with, and the rest of my lab as well, and our funding sources. So just one more thing before I finish. Maria has been spearheading the organization of a discussion group that's linked with the Society for Mathematical Biology cell development subgroup. So the topic is modeling cell development and regeneration, and it's an open discussion group. So if anyone here who is interested in this topic, you can join the Slack channel, and there are regular meetups where people talk about their work. So it's a mathematical biology community discussion group. So feel free to participate. So with that, I am finished, and I thank you for your attention.
Thank you very much, Gary. That was a great presentation. And yes, I'd like to invite questions now. Either put them in the chat or turn your microphone on, and feel free to ask a question. Lucas.
I really like this idea of the transcriptional filter, and I think Maria did some excellent work. I wanted to ask, maybe I missed this, is it that the long genes, there's not enough time for them to get transcribed because RNA polymerase doesn't have enough time to make it to the end of the gene, or is it because the splicing takes a really long time? Like, have you looked at long genes that are spliced and genes at that same long length that don't get spliced, and compare that to the cell cycle duration?
So that's a great question. So we really think it's the first thing, and there's papers that I cited in the talk that show that in kind of early days in cell biology in the 80s, that it's really about if the polymerase can't finish transcribing, you basically have an unfinished transcript, and those transcripts get presumably degraded at the end of the cell cycle. We didn't consider all the other aspects of gene expression, splicing, protein expression in our model, but they would modify each one of those things has its own process and rates, and that lengthens the time that it takes to get from newly processed transcript to actually expressed protein, for instance. And so you could say that it's going to be longer. The whole process is going to be longer than what we model, but in a relative fashion, it's all part of the same general duration, like time scale, or process that takes a certain amount of time, right? So we measured the length the time it takes to generate a transcript. If you measure the whole process of gene expression, it'll be somewhat different. It will be longer. One of the differences that we have with transcripts versus proteins, for instance, or even maybe processed transcripts or finished transcripts, is their lifespan. So unfinished transcripts are thought to be degraded much quicker than finished transcripts, which are usually 3' capped with a poly A tail. Or, sorry, 5' capped, and they have a poly A tail. And also proteins themselves have their own lifespan. So those molecules will survive past cell cycle, like past the cell cycle division, whereas the unfinished transcripts we think won't. Does that make sense?
Yeah, thank you.
something you mentioned, Gary, is that you think some partially finished transcripts might be inherited from the parent cell to the daughter cell. So is there data on that, on how much of it is degraded and how much of it is retained?
So those so yeah, good question. So those transcripts are finished transcripts that are inherited. So once the transcript is finished and it's sort of chemically modified, it's longer lasting. So the chemical modifications like the poly-A tail help prevent the transcript from degrading.
Anmar?
That was really an interesting talk. It's not my cup of tea, but I learned a lot. So I have 3 questions actually. Let me begin with the easiest one. So I do apologize ahead of time because they're really naive. That's not my area of expertise, but so I'm just trying to learn. When you showed us the distribution of the length of the genes, and they look pretty much Gaussian, with that slide. But I'm having trouble reconciling this with your idea of the duration of the cell cycle and the length of the gene. Shouldn't this produce like an exponential or gamma distributions instead, because these shorter genes would get transcribed regardless? Because the cell cycle, if it's long, typically would have minimum length, and it gets prolonged during development. So these short genes would have a high frequency in terms of expression.
Yeah, so first of all, this is on a log scale. So,
Oh, this is a log scale. Oh, I see.
Yeah.
Okay.
Yeah. So sorry.
Log normal, yes.
Yeah, so it's log normal distribution. Yeah. So, but does that change your question? I'm not sure if I think your question still stands. I'm just trying to.
So why do you have a tail? I'm talking about the tail of the distribution that you have for short genes. Shouldn't this be basically overexpressed?
Yeah, so that's what we do see. That's what I was arguing, that we do see that the short genes are more expressed than the longer genes. Is that what you're saying?
Yeah. So basically, yeah. But the thing is, what you're showing in the distribution, this is data, I'm guessing. You have a tail, and actually, the frequency of gene expression decreases. So maybe I'm missing something, but that's how I'm understanding it.
You're saying here that the frequency is.
Well, this is from the simulation. I'm looking at the Gaussian, the log normal distribution.
Okay.
Because you have a tail, for shorter genes.
Oh, in that particular distribution itself.
Distribution, exactly. Like when you go, let's say, to 2, you have a tail here, even though you should be having, based on your idea, you should be having a high expression level of these genes.
Oh, okay. Sorry, maybe I just need to clarify that this is just the number of genes at each gene length. So this is not expression. So I'm missing something. So in general, so there aren't that many. So the highest number of genes is sort of this level here. There are definitely some short genes. There's not that many of them.
But shouldn't these be transcribed at a high frequency?
Yeah, so we think they are. Now, and that's what we're saying, is that short genes are transcribed more than longer genes. We define short genes here in particular ways. So I'm not sure if, you could say that, like,
so you're binning them.
We didn't go to that level of resolution. Yeah, we're binning them. We didn't go to the level of, we didn't create a bin that was just this very, very, very small gene.
I see.
So that's probably what's happening.
What causes that, I see.
Explaining the difference between those things.
Okay, so the other question I have is, in terms of evolution, I would argue that the length of the cell cycle should at least to conserve energy. It would stay stable, but the length of the genes would decrease over time so that you don't actually consume too much energy during transcription. Could that be a potential? I mean, I mean, obviously you're seeing the opposite, but I would argue that at least from an evolutionary perspective, you want to conserve energy, right?
Correct. Yeah. So I think that makes sense for single cell organisms, and just for multicellular organisms is different. So in multicellular organisms, multicellular animals in particular, not plants, from what we can see, there's definitely probably, we would say, a evolutionary pressure to generate lots of diversity in the tissues. And then this mechanism might help do that. Multicellular animal cells don't have the same energy constraints that single cell organisms have because they control their own environment, with like the blood system, for instance. So it's different. It's different in multicellular organisms, but what you're saying, I think, is absolutely correct for single cell organisms. And we do see, in general, bacteria, for instance, have very short genes.
I see. I see. Okay, I think there are lots of people asking questions. I'll keep mine until the end. Right.
Okay.
So I think perhaps Rodrigo.
Hello, can you hear me?
Yeah.
Yeah.
Okay, good. Very nice talk. I actually had one of the questions that you already answered, and the other one, at some point you were measuring the diversity by looking at the cluster number. Would you mind going to that slide again? There was something I probably didn't get, right? It's mainly like a clarification. So is the threshold fixed, or is it always a percentage of it? So my question is, why does it go down after 6 cell cycle duration?
Oh, okay. Yeah. So the main reason it goes down is because the cells, once you allow all the genes to express, they're more similar to each other.
Okay, so
the transcriptome vectors basically have, there's less chance for them to be different because they're just all expressing all the genes.
Okay. So it is because of the clustering.
We think, yeah.
The way that the clustering puts them together, they're more similar with each other.
Yeah.
Okay, so that was my main question, because previously in the other metric that you had for diversity, you had a continuously growing relationship, right?
So you can kind of think of that as two sides of possible ways of explaining things. So just setting every transcriptome to be a different state, if it's even one copy different, is too stringent. Definitely, is one extreme way of defining it, like the maximum level of diversity, and then reducing redundancy kind of reduces a lot of that redundancy and tries to identify a much smaller number of things that you could say are, really truly different by at least a clustering algorithm's definition. And so we get far fewer numbers of things, like, 100,000 versus 100 or something like that. So it's kind of we thought it would be two kind of extreme ways of defining diversity. And what we found is that we found this control-like process happening for both.
I see. Nice. Thanks. And maybe I'm probably gonna get it wrong, but related to what Anmar was saying about the distribution, I was wondering if, Anmar, that would be the case if you had copy numbers as a function of the cell cycle, whereas here I think that they're the number of distinct genes, right? Would that change the shape of the distribution? Maybe, I don't know. It's just a hint, a thought, I guess.
Like genomic copy number?
Yeah, I mean, I don't know, Anmar. Maybe I from your question, Anmar, I thought that you were understanding it as a transcription levels, so that the small genes would also be transcribed very highly. But the number, I think the histogram is not showing the number of copies of short genes, but it's just the number of distinct short genes that there are in the genome.
Yeah, I think.
Well, I thought it was the number. Was it distinct? I don't know, but now I understand exactly how to reconcile these two things. I agree with Gary. It's actually a binning issue, I think.
Yeah, it's binning. Yeah, so we didn't show
rather
this as
than
a continuous distribution. We could have done that.
Okay.
Yeah.
I think we'll take another question from Bianca. if there's some binning questions, maybe some people could also stay afterwards and refine that. Bianca, go ahead.
Hi. I was wondering if you could comment on the directionality. So have you seen studies that look at the other way around, that maybe it's just that because they're shorter transcripts, the cell cycle is faster?
It's an interesting question. Yeah, we definitely think that the short genes are kind of the default because, you know, long genes are more complicated. They have introns, they have many introns. Introns can be very long. It takes longer to generate those genes. So why would you have those genes, as Anmar was asking, evolutionarily? It doesn't make any sense to make something so complicated. But clearly there's some advantage there. So so we've mostly focused on that sort of that direction. If you do study the evolution of these things, you can see gene length changing by introducing introns. And people who study this, they call it the intron delay model. So you can, over evolution, you can change the expression of genes by changing the intron structure. You can make the genes take longer to express and by changing the intron lengths, for instance. So, and there's a lot of changes like that over evolution when you actually compare gene models between species. Having it the other direction where we say genes got shorter is a good question. We haven't focused on too much, but you could imagine that could happen, that there's like a pressure for genes to be shorter to be expressed in cell cycle durations that are very fast. Maybe that's what you're saying. So.
I mean more like shorter genes are expressed more often early in development, and that would make the cell cycle be faster.
Which direction it goes? Yeah, we don't know whether the cell cycle duration was faster or not because of the short genes or vice versa. Yeah, we don't. That's a good question. I think it would be hard to know that, but yeah, we can study that maybe evolutionarily to sort of see how that works. But we need a lot of information about cell cycle durations, which we don't have, unfortunately.
Yeah, no kidding. What drives what at those early development stages? I think at this point, we will wrap it up. If there are people who want to iron out details afterwards, please remain on. But I would like to take this opportunity to say thank you, Gary. That was a really wonderful presentation and really quite thought-provoking in terms of the relationship between cell cycle and how it gets ignored in many of the studies that are done at sort of at a higher scale level. Things to think about. And so thank you so much for coming and presenting today in the seminar series.
Thank you for having me.